All right, so we're live. So thank you everyone for joining this month's installment of EPSP Connects. Today we have Dr. Julie Hope. Dr. Hope is a lecturer at the Scottish Oceans Institute of the University of St. Andrews. Dr. Hope obtained her PhD at the University of St. Andrews and then had postdoc roles and research roles across environmental and ecology groups in Stuttgart, Auckland, and Hull. Lots of interdisciplinary research involving sediment particles, biota, plastics, nutrients, different things. So all related to physical, chemical, and biological drivers that regulate the fluxes amongst those things. So today we're going to learn a little bit more about that. Before we get started, I do want to thank all the volunteers that help make EPSP Connects a reality. People, that reach out to our potential speakers, panelists, etc., that help us run this, keep the website updated, everything running. Today joining me is Mareike, who will help with questions in the end. We're also live on YouTube, so if you're watching the live stream, we'll keep an eye on that as well to answer questions as they come. If you have anything to add, Mareike, or we're good to go.
No, we're good.
All right, Dr. Hope, the floor is yours.
Thank you, Roberto. Thank you, Mareike. As Roberto said, I'm a lecturer at the University of St. Andrews, but today I'm going to present a mix of different pieces of work that I've done while at the University of Hull in the EEI, the Energy and Environment Institute, and also here in St. Andrews. So I'm going to start with a little bit of the basics of what is a biofilm. So a lot of my work has been focused around these biofilms that are on the sediment surfaces and coastal systems. And really, these are a mixture of different organisms. So you've got microphytobenthic primary producers in there, you have bacteria, and they're in this matrix of extracellular polymeric substances. And this is largely composed of sugars or carbon and proteins, and also the microorganisms within them. Now they form in many sediments, but also, sediments, but also many other surfaces. So they form in the inside of things like pipes. We have biofilm in our teeth. We have biofouling on boats. And it's this same sort of sticky matrix that we have. Now, these can vary visibly from a kind of brown hue on the sediment surface to really thick mats that are completely covering these sediment grains, as you can see in this lower image. And you can see in that top black and white images, these microorganisms, the microphytobenthos, are tiny, tiny little microscopic primary producers, and that is a single grain of sand in the center. So individually, they are very small, but they collectively create these large mats, and they play a really important ecological role that we'll go into in just a second. So you can see here, again, if we take something like glass beads, and this is a low temperature scanning electron micrograph, we can develop a biofilm over them, and it creates this carpet of EPS, that kind of fluffy material that you can see, and you can see the little shapes of some of those diatoms in there as well. And what this does is it binds these particles to each other and also to other things. So those microphytobenthic, the microscopic primary producers that I was talking about, are really important, and they underpin a variety of different processes in our coastal systems. They underpin coastal food webs, not just their benthic food webs, but pelagic. They promote biodiversity in coastal sediments, and they play an important role in nutrient cycling and obviously that primary productivity. But they also play an important role, as you can see in that bottom left-hand side, in this biostabilization and habitat formation. But as I've developed from my PhD, I've been realizing that they're part of this larger consortium. Today we'll focus on biofilms, and it is those microorganisms, but they are part of these larger communities. So the microbes, the bacteria, the algae, and the fauna all work together to influence these processes. Now, as I said, my PhD was really focused on biostabilization. So how do these biofilms form on sediments, and how does that affect things like sediment transport and sediment deposition? But now I'm increasingly expanding into all these different processes, and actually those processes in the bottom there in the light blue are all really well linked. So when we have more biodiversity, we have changes in nutrient cycling. When we have primary productivity, we have effects on biostabilization. When we have removal of sediment, it affects the nutrient cycling as well. So this biostabilization can protect the underlying sediment from erosion and movement. And what that means is that more energy is required to move those sediments via suspension or bed load transport. If you think about that bottom picture there, it's creating like a carpet. Eventually it will have enough force from flow to remove it, but while it's still there, it protects that underlying sediment. And these biofilms are also sticky, so they can capture and retain various other particles. That might be other things like sediment grains, but also pollutants and other particulates that settle out from suspension. And that's why I've been starting to look more at these benthic pelagic coupled processes. It's more than just about the exchange of sediment. When flow disturbs the sediment, it releases things like carbon and nutrients and those microphytobenthic cells into the water column, and that affects things like water quality. And it can also have a release of contaminants. So you think about really muddy sediments, they might have a lot of heavy metals within them. When flow is disturbing that, those metals become are more available in that water column. And we also have to remember that flow brings in those things too. So it brings sediments to the intertidal environment, it brings in nutrients that help things grow, but it can also bring in contaminants. So the first thing that we're going to look at today is the role of biofilms in trapping microplastics. And this is going to be the main thing that we speak about today, and I'll only touch on some of the others. But these biofilms that are on the sediment surface, we don't really know a lot about them. We know there's many ways in which biota can interact with plastics even before they settle to the bed. You've got things like biofouling. You have those biofilms forming on the surface of particles in the water column, and that can change the settling dynamics. You've also got things like ingestion from larger organisms, and then they can repackage them into feces, and that can help settle them to the ground. But these sediments are the ultimate sink for plastics. Now we're starting to understand how those plastics on the bed can interact with different benthic organisms, so they can get entangled in things like seaweeds or seagrass. They can be buried by bioturbating fauna, and they can be eaten by those benthic feeders, those fish and other organisms as well. But what we know relatively little about is that exchange of plastics across the sediment-water interface. Often when we go out and sample sediments for plastics, we do it at low tide in the intertidal environment. We only get a snapshot of what's there at that time. We don't know how they're fluxing in and out of the sediment and into the water column. So there's two aspects to the talk today that is really, I want you to think about, is really that these plastics are known to trap and bind to other small particulates. So they have the potential to trap plastics into those biofilms as well. They're also a vector for many other contaminants, and the plastics themselves can also have toxic chemicals within them. So they have the potential to embed themselves into those biofilms, but they may also adversely affect the biofilms and other benthic organisms. Okay, so what do we know about plastic transport already? So the resuspension dynamics of different plastic polymers was investigated a few years ago by Waldschlager and Schuttrumpf. And what they developed was this relationship between the ratio of the Shields parameter of the plastic and the sediment bed, and that against the diameter of the plastic and the sediment bed as well. And they did this for a variety of different sediment grain sizes and a variety of different plastic polymer types and sizes. And they generated this equation at the bottom, which it fares quite well. It's got this nice linear relationship. It's a good approximation, and given the wide range of different sediment grain sizes and plastic sizes and densities, it works quite well. And what this is saying is you could predict that microplastics erosion threshold if you know those different parameters, if you know the grain size and the the plastic grain size as well. But what this is missing is the fact that there's no cohesive fraction. The smallest grain size that was considered here was 450 microns. So it's non-cohesive sediment. There's no clay particles in there, and there's also none of that biological cohesion, so those biofilms. So what is the influence of biofilms on these processes? Well, we know that the EPS and the biofilms are sticky by nature, and we know that they can trap sediment particles on the bed. So understanding the role of these biofilms in plastic transport is important for understanding that transport from land to sea. But if those plastics are retained on the bed for longer times, it means that they have the potential to have larger ecological effects on benthic organisms as well. So we could hypothesize that benthic biofilms might increase the shear stress required to resuspend plastics from the bed. The biofilm is more sticky than a non-cohesive sediment bed. And we can perhaps hypothesize that if you have more biofilm present, you're going to have more plastics present. So we're starting to think about whether you can see these as little hotspots of plastic accumulation. Now, I was also interested in the influence of different associated contaminants. So we know that these plastics pick up different contaminants as they're transported through the water column. So we wanted to investigate that also. And we did this through quite a small scale study where we had 36 different mesocosms. We subjected these to different conditions. So we added nutrients to promote biofilm growth. And in the control systems, we actively killed the active biomass that was there. So we're killing off the microphytobenthos and the bacteria. Then we had a low biomass level and a high biomass level. And we put these in a tidal simulation for 21 days. They're not under flow, but they were able to emerge from the water column to allow those plastics to settle on the surface. And then during high tide, those plastics would begin to float in the water column again. Now we used polyamide particles. These are 500 microns. They were exposed to different heavy metals. So we used copper and lead as two representative contaminants, and we fluorescently stained them so that we could track their movement. Now, this little inset image on the middle right, you can see it's a little petri dish, and we embedded that into the mesocosm so that we could remove that later to conduct some erosion measurements in a small flume, which I'll show you next. And those particles were added on day 14. So we allowed the biofilm to develop. Then we added the particles, allowed them to interact with that sediment bed over a week. And then we started to look at the different measurements. So we conducted various biochemical analyses, and we looked at surface sediments with a small, these 6 small syringe areas. And then we were able to pop out that larger circle. That was our inner petri dish, and that inner petri dish was able to be transferred into a flume system. Now today I'm only going to look at chlorophyll A analysis, but we did look at grain size. We have DNA to look at the bacteria and the diatom community, and also microscopic ID of those diatoms to see if the community composition has a role in how sticky these biofilms are and the trapping of plastics. So today I'll focus on the chlorophyll A analysis. It was the main driver that we were interested in. So that small petri dish, as you can see, was placed into a small benchtop recirculating flume. Those of you that are more in the hydrodynamic side might be horrified by this. It's a very small scale. It's only 1 meter by 1/4 of a meter. It has a false floor. We were able to put those little petri dishes in flush. We were able to squeeze in an ADV downstream, and we were able to incrementally increase the flow over time. We were only able to increase this up to 30 centimeters per second, just again because of that small scale. We couldn't exceed that. Now we had 2 DSLR cameras. We had an aerial camera which was facing down onto that core, but unfortunately the vibrations from the table were making that quite difficult. So we really only got before and after pictures of those cores. And then we had this side camera that you can see in the image where we continuously recorded the flume run throughout. So we filmed these under UV light to fluoresce those plastics, and that allowed us to see the movement of the particles over time. And what we did was these are two of the aerial photos. We were able to look at the loss of coverage in those particles from that circular area, which is the Petri dish, using loss of coverage in a MATLAB code. I just want to show you two videos of the first flush of the plastics being removed. So you can think of this as tidal inundation. Now these cores were flushed. They were all put into water to remove any loose particles. This is the water encroaching on a core that has no biofilm present. You can see that those particles are just being washed straight off. There's some that remain, and then we can see how those remaining particles are removed over time. And then hopefully you can see, sorry, my editing skills aren't great. When you see the biofilm is present, you can see when that water's encroaching, those plastics are really embedded onto that bed. They're sticking to that biofilm and they're not being removed by that encroaching water. Okay, so we wanted to look at different biomass levels. So this plot's just really to show you that we increased those biofilm levels successfully. We have some background chlorophyll that's still there, even though we killed off the active biofilm. You have that low level at the bottom. So the white boxes here are the control, no biomass treatment. And then the light green are the low biomass and the dark green are the high biomass. So you can see that we increased the biomass levels, and across the bottom we have our control sediments that had no metal additions, and then we have plastics that were exposed to copper, and then we have plastics that were exposed to lead. Now we didn't see any significant effect of those metals, but you can see there in that far right, the addition of the lead really makes those chlorophyll A levels more variable. Now, chlorophyll A is a proxy of that biomass that's there. So we're getting a more patchy biofilm when we have lead there. And just a reminder for those that are not familiar, when we have a higher microplastic erosion threshold, that means that the plastics are harder to resuspend from the bed. So when we look at the control, no metal additions, just these first three boxes on the left, you can see that we have a really low microplastic erosion threshold when there's no active biofilm present, that white box. You can hopefully see that that is substantially increased when we have a low biomass biofilm. The erosion threshold is much higher. It's much harder to remove those plastics from the bed. But then we also seen this reduction in the erosion threshold at a high biomass level. Now, I mentioned earlier on that we didn't grow these biofilms under flow, and we know that the biofilm is stronger and more compact when you grow under flowing conditions, but we tidally inundated them in order to keep those plastics where they were. So what we think is this biofilm in the high level is actually growing really fluffy. If you think of it as very kind of fluffy and loose, and that means when the flow is coming across there, it's easy to rip up that biofilm, and then those plastics go with it as well. And then if you move to these copper and lead treatments, again, you can see that the treatments where we have biomass are much higher than the white boxes. So the green boxes are all higher, but those control sediments are all very low, and there's not much difference or effect, except perhaps that in the lead treatment you have much more variable erosion thresholds coinciding with that variability in the biofilm. So we wanted to compare our empirical values with the theoretical microplastic thresholds derived from that equation in Waldschlager and Schuttrumpf. And what we found is that even when we collapse out the metal treatments and we're just looking at the amount of biofilm biomass that's there, we don't see any significant difference between the theoretical value, which is that dotted red line, and our control thresholds. We see quite a good correlation between them, and it holds quite well. But hopefully what you can see here is when we have biofilm present, those erosion threshold predictions or the theoretical values aren't falling in line with that prediction. And this is regardless of metal contamination. I've given you just it collapsed here, but when we look at them as individual copper, lead, and control treatments, we've seen the same throughout. So we were interested in understanding when those plastic thresholds might be surpassed in an estuarine or an intertidal environment. So we put those microplastic erosion thresholds into a hydrodynamic model for the Humber estuary. And when we looked at the whole estuary, we've seen that those microplastic erosion thresholds were exceeded about 76% of the time over the tidal cycle when there was no biofilm present. But when there was a biofilm present, those erosion thresholds were only surpassed about 36% of the time. When we reduce that to just the intertidal area of the estuary, we can see that the plastic thresholds were exceeded about 11% of the time when there was no biofilm present. But again, that was substantially reduced to 4% of the time when we have biofilm present. So what does this mean? Well, benthic biofilms are increasing the microplastic erosion threshold. So the presence of those biofilms is making it harder for those microplastics to be resuspended in the outgoing tide. And it is above those theoretical levels that we have in place at the moment by some studies. And that biofilm presence can reduce the amount of time in which the plastic thresholds are exceeded over a tidal cycle, at least for the Humber estuary where we've looked at it. And that means the plastic retention in these estuaries is really going to be driven partially by those biofilms that are present, and they really grow over summer periods. So when there is a lot of sun, you have a lot of biofilm. Those plastics might be held on the bed for longer, and that might interfere with their transport from land to sea. Whereas in winter, when you don't have a lot of biofilm present, there may not be as much of an effect of this. And this doesn't only affect the transport of plastics from land to sea, but it will affect things like plastic burial and the effect it has on different processes within the bed and how it interacts with different benthic organisms. So when we have plastics on the bed, we have fauna that are feeding on the biofilm. That means that they're going to be ingesting those plastics, and as they bioturbate through the bed, they're going to be burying them into the deeper sediment layers as well. So what's next? So we're in the final stages of this paper. We're looking at some of the DNA analysis at the moment to see whether the community composition of these biofilms plays a role in the mediation. And this is really to try and get at the differences in those metal treatments. Are the metals stressing and creating different communities, and does that have an effect? So we're hopefully going to get that out in the coming weeks. And my details are there for that study. But what I want to move on to here is that once those plastics are trapped on the bed, they don't just sit there, they can affect the biofilm community and how it functions as well. So these biofilms are really active sites for nutrient uptake from the water column, and they're important for biodiversity. They're also a food resource for many animals. So first of all, we'll go back to that sediment stability. So if those plastics are accumulating in biofilm, does it affect the biofilm community? So we added fibres and pellets of polypropylene to see if there was a difference in how they affected the biofilm community. And we found that, again, that chlorophyll A level is substantially reduced with both fibres and pellets. And these are high and low treatment applications. So we have a decrease in the amount of microphytobenthic organisms there. And then we measured sediment resuspension using a cohesive strength meter. And what we found is that stagnation pressure, the amount of force required to resuspend particles, was again reduced with the presence of those plastics. And we had a greater effect of the higher concentration of pellets, which is a little bit difficult to explain. You would think perhaps the fibres would have a greater effect. But what we can see from this is that those plastics can adversely affect the biofilm biomass and how those biofilms can stabilize the bed. Okay, so what are the other effects of microplastics on these different beds? Just want to reiterate the different processes. So as I said earlier on the right here, biofilms can influence those sedimentation and resuspension dynamics. And that's not just of those sediments that I just spoke about, but also particulate organic matter, so the POM. But they also play a huge role in sediment metabolism. So they are primary producers, so that gross primary productivity is influenced by them, and also that respiration. And it's not only those microphytic benthos, but also those bacteria in the bed and the fauna that are respiring. And then they also play a role in this nutrient exchange across the bed. So they're also important for different exchange processes. So these diffusive fluxes of nitrogen and phosphate on the left-hand side there are influenced by the presence of these biofilms. And what we were interested in is whether any of these other processes are also influenced by the presence of plastics on the bed. So I'm only going to show you a few different snapshots of what we've looked at in the next few slides, but hopefully it can show you that they do have an effect on different processes. So first of all, we're going to look at whether those plastics on the bed can affect oxygen dynamics, so the primary productivity of the biofilm under different light conditions. So in terms of light, we're talking about if you have a more turbid system and your light is reduced on the bed, you perhaps you would expect there to be less primary productivity, but is that compounded by the presence of the plastics? So in ambient light, this is about 200 micromolars. You can see that we do get a reduction in the net primary productivity when we've got fibers and fragments present on the surface. These are in the surface 1 centimeter of the sediment. And then you can see that anything positive, anything above 0 is oxygen production out of the sediment bed, and anything that's negative is an uptake of that oxygen. So how much drawdown or respiration there is. And hopefully you can see that when we shift from high light to low light, we get a shift from net O2 production in the yellow bars to net O2 uptake. And what we're seeing here is actually we have a greater effect of those plastics when we see it in the high light conditions. So under low light conditions, that's the dominant factor. The plastics aren't having as much of an effect, but under good light conditions, those plastics are affecting the environment. So what this is saying is if we get an increase in turbidity, it could influence different net primary productivity, but that might be altered by the presence of plastics. And then we've also looked at plastic effects on oxygen production, again, from these biofilms under different heat conditions. What hopefully you can see again here, that 0 line is, you can think about that as your sediment surface. Anything below there that's negative is that oxygen being drawn down into the sediment. And anything above that line is oxygen being produced from the biofilm in the sediment and out into the water column. And hopefully what you can see is under ambient temperature conditions on the left here, you have, this is a different system by the way, you have net oxygen uptake, and there's a difference between the control sediments, which are the white boxes where there's no plastics presence, and when we add plastics in the blue boxes. But you see a shift in the way that these are metabolically behaving when you've got plastics present. So heat spikes were multiple hot days, and you can see that plastics shifted to be a more productive environment, which at first may seem like a good thing, but we have other nutrient data that I've not presented here. But what I want you to see here is that when we have heat spikes, which is multiple hot days versus a 5-day heat wave, you can see that those directions and trends are actually completely different. So under control in the end there, you've got high productivity under heat waves, so the temperature is stimulating the biofilm, but when the plastics are there, it's not. it's still a net uptake. And that is down to greater respiration with the heat spike. So that heat is stressing those organisms within the bed to a point, and it's shifting that metabolism of the benthos. So don't want to go into this in too much detail, but it is shifting the way that these are metabolically behaving and the productivity in these systems. And then again, that'll have an impact on how those systems behave. So what's the take home here? Well, biofilms can modulate the fate of contaminants such as plastics, but actually the very biofilms that help trap those plastics might actually end up functioning really differently because of them. And biofilm changes, not just due to the plastics, but other stressors in the environment, can alter the role in these different pelagic, benthic pelagic coupled processes. So things like nutrient exchange may change, the system metabolism, and also the resuspension of different sediments and particulates. And what I want to stress is that these processes are intricately linked. So the ecology in the bed, the biochemistry, and also that physical environment are really important. And I want to mention that I've not really talked about anything with the fauna here, but we have considered the fauna as well, because they do play an active role in some of these processes as well. And these biofilms really link those hydrodynamics and the transport of sediment and contaminants in these coastal and estuarine environments. So I want to thank, I've got a long list of collaborators across these different projects. Hopefully all the papers will be out shortly. I've had a few interns who've painstakingly done some diatom identification on the microscope. And I want to just thank the funders for those different projects as well. My details are in the corner there if you'd like to get in contact about anything and potential future collaborations, because some of this work is something that I would like to take forward and will be proposing later in this year. Thank you.
Right. Thanks, Julie, for your very interesting talk. If anyone has any questions, feel free to type them in the chat, or I guess the group is small, so you can also open your mic and ask the question. While everyone is warming up, I will ask something. I was mainly wondering how you mentioned that you didn't brief on it, but how much this biofilm varies between different areas, and how much impact the different impact you would have in different environments. Like, what is like the range you would expect?
So yeah, the biofilm growth can vary substantially, even within an estuary. It is very dynamic and patchy. Those later plots that I showed where we were looking at the productivity, we've found that even down in Hull and Humber, the sediments were net phototrophic or autotrophic. There's more productivity than's being respired. So you have oxygen being produced more than it's being taken up. But even as I've moved north to Scotland, perhaps because of the weather or the climate, those systems are net heterotrophic. So those organisms in the bed are actually drawing down more oxygen than they're producing. And again, that can be the microbes, the fauna, and the microphytobenthos. So you see a really quite a substantial difference in how the biofilms are functioning. And that's also in terms of how they stabilize things or how sticky they are. So different stressors, different flow conditions, all of these variables vary the thickness and how strong that biofilm would actually grow as well. I don't know if that answers your question in terms of quantifying it. You could quantify the thickness of it, but we've done some studies where we know that growing them under flow conditions actually strengthens them because they grow more compact, and they're not protruding up to kind of into that benthic boundary layer. So when you increase flow, they're not ripped off quite so easily.
Thanks, Julie. Oh, Andrew has a question. Go ahead.
Yeah, it's kind of a similar question really. So I was just interested in this variation in the biofilms, but maybe how do they vary as you go from like fresh water into saline, as you go through the kind of
Yeah.
mixing them? Do they change a lot? Or
Yeah, so there's been some previous studies by Spears, I think in the early 2000s, where if you do that gradient down from fresh water into saline water, you get a change because you've got that different ion strength, and it actually binds the biofilm really differently. So you get stronger biofilms as you come downstream, but it can be a gradient as you come down an estuarine environment, for instance. So
yeah,
all those chemical properties, all the inputs of things like nutrients. So if you get loads of nutrients coming from the catchment, it might um, increase that productivity, but in terms of the strength of it, we're not entirely sure.
yeah, no, interesting. And is there much on like looking at some of the emerging contaminants and the role that they have? So like
Yeah,
around some of the kind of organic contaminants, microorganic contaminants, like I don't
yeah.
know.
So there's been a few. I put in a proposal last year. It wasn't funded, unfortunately, but trying to look at PFAS
Yeah,
because we've got a lot of PFAS that are coming down our watersheds as well. And again, we know on a kind of microscopic scale in a petri dish, we know that they would have an effect on these organisms, but we don't know the effect on the function of the organisms or the biofilms themselves. So it was something I tried to get funded last year, but it never got through. Um, but there's a variety of different contaminants, and we can do these experiments looking at a variety. In fact, I think it's my next slide. I look at different things like nutrient inputs, climate change, plastics, sediment concentrations, and storms. All of these things are really happening at the same time in our estuarine and coastal environments. It's not just a single stressor, and I think that's why it's really important to think about how they're interacting as well.
yeah, yeah, absolutely. Great. Thank you. Thanks, Julie.
Thanks.
Got a question in the chat from Stephanie Donor. Julie, wonderful and interesting work. Have you looked into deeper depths? In parentheses, 20-meter and deeper.
Yeah, so I've kind of worked in both extremes and not a lot in the middle ground. So I do a lot of my work interested in the intertidal, but we've done some work where we've looked at the stabilizing effect of EPS that forms these biofilms in turbidite systems. So when there was the Kaikoura earthquake in New Zealand, and we've seen these kind of mudslides in deeper waters, and we can see that that EPS is having an effect on those systems as well. We've done a little bit more on the carbon side in the more inshore environment, but those deeper levels. But there's, as long as you've got a photic system, in the southern bight, it's been shown that these biofilms are really active even down to 180, 200 meters. So if you've got light penetrating through that water column, you've got these biofilms producing. And you have a lot of bacteria in the bed that can also contribute to these biofilms. So even if there's not a lot of microphytobenthos, you still have a lot of this sticky biofilm that's there on the sediment surface, even down to 200 meters.
All right, I have a question of my own. From people dealing with sediment transport, and you showed your shields diagram, and there's interest in, okay, what do we do in terms of initiation, or how do we deal with removing the biofilm before we can consider eroding the bed?
Mhm.
That's kind of the mindset, and then we want to parameterize the biofilm as we would cohesive sediment. We need some kind of erodibility or some kind of coefficient. Have you looked at any of that? That's part one, I guess. And the second part is this, you've alluded to this, there are multiple communities that conform these things. Is the chemistry really that different? And is the stickiness specific to the local chemistry, or they're all as sticky?
So I'll start with the second part first. I think the environment and how stress, so like copper can actually initiate more EPS production because that EPS forms a barrier and it can actually protect the biofilm. So some stressors actually stimulate EPS production, but whether that EPS is stronger is a different story. So we know that copper can chelate with the EPS and it can bind differently. So I guess that there's a little bit more to do in terms of those chemical bindings. We know that EPS is largely negatively charged, and that depends on the charge of different chemicals, or again with the ionic strength or different pollutants, how those interact might vary. And again, that will affect how strong they're binded together, how strong they're binded to different sediment grains or different pollutants. But it's so variable. It's really hard to give a single number to the effects of the biofilm. But then coming back to the first part, we stuck these numbers into a hydrodynamic model to see, okay, when would they be surpassed? But the aim for a project that I'm hoping to submit at the end of this year is really thinking about how can we use different ways of looking at the biofilm on a larger scale. We had to do this small scale to take little mesocosms, because it's really hard to grow a full biofilm in a 15-meter flume and then erode it in seconds and then grow it again for 3 weeks. But if we can start to use things like remote sensing, and we have different indexes where we can quantify either the health or the amount of biofilm across a whole estuary, then we might be able to kind of parameterize better where those hotspots might be, but maybe how likely plastics are to be settling and sticking versus settling and resuspending straight away with the tide again. I don't know if that helps.
Yeah, thanks. It's interesting, lots of work to do. These things are patchy, so it's not a single value for the whole estuary. But there's another question, Mareike.
Yeah, there's another question in the chat from Stephanie Donner. So she's asking, how do you feel about the use of xanthan gum as a surrogate for EPS in laboratory studies of sediment transport impacts?
I've used it, yeah. So it's super strong stuff, but it gives us a really nice way. As I said, if you're trying to grow a biofilm in a 15-meter flume, or we've done some work at the Deep at the Total Environmental Simulator, which is 10 meters by 6 meters. You need a proxy. You can't grow a biofilm for 3 weeks till it's fully developed and then sweep it away within seconds and then have to reconstitute that bed and grow it again for 3 weeks. We just wouldn't be able to do it. So xanthan gum's a really good proxy. It works. It's quite strong. It's quite potent, and it really doesn't give you. It doesn't give you the effects of those different communities. It's just a kind of one EPS. It's a proxy for the extracellular polymeric substances. But you could start to mix it with different things and start looking at different compositions of the xanthan gum. So I'm all for it. It doesn't give us a full picture of the structure of the EPS. Like I said about the biofilm growing really fluffy under stagnant conditions, but really compact under flow. You just don't get that with xanthan gum. You're kind of mixing it in and it becomes very gel-like, but it has a place in order for us to upscale things and for us to do those replications that we need to try and understand things, I think. But I'm interested to hear what other people think. Maybe people disagree.
Yeah, it was a bit of a setup. Sorry, I've got my camera on now. Hello, everyone.
Hi.
No, this is wonderful. And it was a bit of a setup because I'm at the Naval Research Laboratory in the Sediment Dynamics section.
Okay.
And I had a PhD student work on, we have a smaller flume as well. It can do unidirectional, combined, and oscillatory conditions. And we cultured diatoms both with predatory worms and without. Mm-hmm. And kind of looked at that erodibility with the community impacts, and compared it to xanthan gum and clean sands too. And our findings were that the xanthan was really no comparison, at least in that
because
regard.
of the strength of it, because it's that high strength. Yeah. I think as well, when we think about so these diatoms that are coming up to the surface, they're really producing it in those top microns of space, but often when we try and mix it into flumes, we're trying to mix it through a greater depth because it's not physically possible to smear a layer on like a biofilm.
Mhm.
So then, yeah, you have a kind of order of magnitude difference in the effect that it has. So I don't think it's not comparable to the role of diatoms. But if you're thinking about, we've done it for larger scale projects where it's looking at how small amounts or the relative amount of that xanthan or EPS can affect bed form development. That it's not really comparing it with the diatoms, but it's kind of saying it is if you have more and more EPS, it has this dampening effect on the bed forms. So yeah, it definitely has its place. I agree with you, it's super strong. I remember I used it in my PhD and I tried with a 1% mix and I ended up with solid pots of things that I just couldn't get the sediment back out the pot. And it was like, okay, and we'll move that to the side and go on to the next one. But yeah, it'd be really interesting to see the work that you're doing. But this is where, like you say, those predatory worms, we need to start building up that complexity. And it's this kind of balance because perhaps the sediment or the hydrodynamics folk or the modelers want it simple enough, but also it is a complex ecological system. So it's finding those balances between having a nice proxy. And I think, like we said with the xanthan gum, trying to get that into the shields diagram and trying to understand, okay, what effect does biological cohesion have? That's far easier to do with a proxy than it is with a biofilm that in itself is really complex. And then you've got animals and bacteria and everything else that's going on, unfortunately.
No, I agree. I won't dominate this, so I'll email you outside of this meeting.
Yes,
Thank you
I'd
so
love
much.
to hear from you. Yeah.
Yeah.
Thank you.
All right, so it looks like we have no more questions. All right, well, our speaker for November emailed me this morning saying that something came up, so we've pushed them until probably May, but don't worry, we'll find someone and let you know through our typical channels, social media, and mailing lists. There will be someone in our EPSP Connects for November. Thanks again, Julie, very much. And look forward to learning more about your work in the coming weeks. Thanks, everyone, for joining.
Thank you so much, everyone. Thank you.